Hello friends and welcome to my channel. In this lecture we are discussing the bacterial transformation. So please subscribe my channel Easy Life Science, like my video and share the video to your friends. So let's start. The bacterial recombination. The bacterial recombination is a process in which a recombinant chromosome is formed from a DNA molecule derived from two different parental cells. In prokaryotic organism, a small portion of a genome from one cell, that is a donor cell, is transferred to the another cell, that is in a recipient cell. The portion of a chromosome transferred from donor cell may combine with a recipient chromosome to form a recombinant chromosome, and this recombinant chromosome is an incomplete zygote, so it is called as a merozygote. The original genome of the recipient cell is known as the endogenote, and the portion of a genome transferred to recipient cell is called as a exogenote. The recombination process is not a obligate process in a bacteria, but it occurs in a certain cases, or it occurs occasionally. The recombination process may be transformation, conjugation and transduction, and these are also called as a HD HGT, or a horizontal gene transfer, transformation, conjugation and transduction. In a transformation, cell free or a naked DNA which contains a limited amount of genetic material is transferred from donor cell to a recipient cell. In an image you can look for this donor cell and in transformation, cell free or a naked DNA which contains a limited amount of genetic information is transferred to a recipient cell. In conjugation, for this transfer of a genetic material there is a need of a plasmid, so it is plasmid mediated recombination. And in transduction there is a need of a bacteriophage for a transfer of genetic material from donor to a recipient cell. So these three are HGT, horizontal gene transfer, and the cell that can be used for a transformation are called as a competent. The transformation process discovered by scientist Frederick Griffith in 1928. It is discovered in a bacterium, that is in a Streptococcus pneumoniae, and the scientist Griffith suggested that the bacterial strains are capable of transforming themselves by some factor, and this factor is called as a transforming principle. After some time, the three scientists Avery, McLeod and McCarthy, they demonstrated that transforming principle is nothing but a DNA, and which giving evidences that is a DNA is the genetic material of a bacteria. The transforming principle, we know that it was discovered after a Griffith experiment, especially the fourth step of a Griffith experiment. If you want to study it, in my channel there is another video, its link is in a description box, so go through it and observe or watch the Griffith experiment, especially the fourth step where the mice were injected with a heat killed strain of bacteria plus a R strain, and at this step the mice were get killed That means heat killed a smooth strain, transport to a R strain, and R strain become virulent, and this process is called as a transformation. And the agent that was responsible for transformation was called as a transforming principle. The transformation occurs naturally or it occurs artificially. The natural transformation is observed in gram-positive bacteria like Streptococcus, Bacillus, and Thermoactinomyces. The natural transformation occurs in some gram-negative bacteria like Azotobacter, Haemophilus, Acinetobacter, Moraxella, and Pseudomonas. The transformation event is having three major stages: development of competence, DNA binding and uptake, establishment of donor DNA in a recipient cell. The first, that is development of competence. The ability of bacterial cell to take the DNA from a medium is said to a competence. The competence can be increased by a physical or chemical treatment. Competent bacterial cell can take the DNA with a size less than 15 kilobase pairs. The competence may be transient or it run for a few minutes in a Streptococcus, while it lasts for more than 1 hour, for example, in a Bacillus. The scientists Notani and Setlow in 1974 described that the mechanism of a bacterial transformation in Streptococcus pneumoniae. The competent state is transient and it persists for a short period. The competent state can be induced by a competence activator protein. The competence is an ability of a recipient cell to take up a foreign DNA through the changes in cell wall. This changes involves the formation or activation of a special DNA receptor proteins, and these special DNA receptor proteins that can be induced by a small protein, and these small protein are called as a competence factor. In a competent Streptococcus pneumoniae cell, about 30 to 80 cell wall receptor sites are capable of binding the double-stranded DNA or double-stranded donor DNA, whereas in a bacterium, that is a Haemophilus influenzae, the Haemophilus influenzae possesses 4 to 8 receptors, and these receptors can recognize a DNA of a 11 base pair sequence. The competence factor binds at a site on a cell surface and it stimulates a production of 8 to 10 new proteins which are required for a transformation. Some of these proteins includes atolysine, DNA binding protein, and a nuclease. The competence is not a permanent feature and it occurs only at a certain times in a life cycle of bacteria. The duration and a time of occurrence of a competence varies with bacterial species and growth conditions. The competence usually develops at the end of log phase just before the tertiary phase. In Streptococcus pneumoniae, development of competence requires 90 to 100 minutes after beginning and it lasts for 7 to 15 minutes. But in Bacillus it requires several hours. The changes induced by a competence includes increase in a porosity of cell wall, increase in a atolytic enzyme activity, and increase in a average length of a chain of a cell. Most changes occur along the equatorial region of a cell where the new growth is formed. The competence factor accelerates the process of transport or leakage of attenuation molecule into a periplasmic phase. The structural changes in a competence cell induce a numerous vesicle, that is a transformosome or transformosome buds. These are formed on the surface and it contains protein, and this protein facilitates uptake of transformed DNA. This is a diagrammatic representation of a transformation in a Streptococcus. This is a recipient genome, or it is a endogenote, and on a cell wall there is presence of activator binding protein, and activator binding site is there, where to the outside of a cell the double stranded DNA molecule get converted into a single stranded DNA. One strand get degraded with the help of endonuclease, and further only the single stranded DNA of a donor cell is get entered into a cytoplasm of a recipient cell. Now the events which occurs in a transformation, the first, that is a DNA binding as a result of a random collision. The donor DNA comes contact with a cell surface of recipient competent bacteria. The first, the DNA binding is reversible. It takes 4 to 5 seconds. Thereafter, this process of binding of DNA becomes irreversible for about 2 minutes. It remains in a non-transforming state. Thereafter, before 5 minutes, it is converted into a transforming state. The period of 10 minutes during which no transformation occurs in a competent recipient cell, this period is called as the eclipse period. Both type of DNA, transforming and non-transforming, binds to a cell surface, and on a cell surface of a donor cell there is a receptor site. But in case of Haemophilus influenzae, there are the transformosome buds, and these transformosome buds are formed on a surface which contains protein, and that mediates the DNA uptake and penetration. In Streptococcus pneumoniae, the competence factor induces the ability to bind DNA molecule. Then after binding a DNA molecule to the recipient cell, next event is a penetration. The DNA molecule that binds permanently to the recipient cell and enters in the cytoplasm of recipient cell. This DNA which is entered into a recipient cell is resistant to the DNA's degradation. The nucleolytic enzyme located at a surface of competent cell acts upon a donor DNA molecule when the donor DNA molecule binds to a cell membrane. Then the enzyme endonuclease first of a recipient cell. This enzyme is associated with a cell membrane of a recipient cell, and this enzyme endonuclease first acts as a DNA translocase by attacking and degrading one strand of double-stranded DNA of donor cell. Consequently, only the homologous single strand of a DNA enters into a recipient cell, and this is confirmed by performing an experiment by radio labeling of a donor DNA. In organism Bacillus subtilis, the degradation of one strand is being delayed, and hence here both the strands enters in a recipient cell. Successful transformation occurs with a donor DNA of a molecular weight between 30 lakh and 8 million Dalton. While increasing a concentration of a donor DNA, the number of competent cells can be increased. After penetration, the donor DNA into a cytoplasm of a recipient cell, it migrates from a periphery of a cell to the bacterial chromosome, and this movement is different in different bacteria. Then the next event is a synapsis formation. In this synapsis formation, single stranded DNA that is entered into a recipient cell is coated with a SSB protein, that is a single stranded binding protein, which maintain a single stranded region in a replication fork. The single strand of a donor DNA or a portion of it is linearly inserted into a recipient DNA, that is a integration. Then the recipient protein or recipient bacterial protein like RAC A, for example, in a E. coli, this RAC A protein facilitates a DNA pairing during a recombination, and this protein RAC A causes local unwinding of double stranded DNA of a recipient cell from 5' end. The base pairing, that is synapsis, occurs between homologous donor single stranded DNA and a recipient DNA. The unwinding of recipient DNA continues at the end of assimilated DNA, and it allows a fraction of inverting DNA to increase base pairs, and this means branch migration. Then the last event, that is integration. The endonuclease cut the unpaired free end of a donor DNA or a recipient DNA, and this process is called as a trimming. Then the nicks are sealed with a DNA ligase, and because of sealing the nicks, there is a formation of a heteroduplex. And the heteroduplex after replication of heteroduplex, the homoduplexes are formed. One is a normal type and another one is a transformed type. The normal does not form the characteristics of a donor cell, whereas a transformed duplex forms the characteristics of a donor cell, and these two cells or these two duplexes are differentiated through a plating method by using antibiotic markers. In a transformation, competence is very important. Competence means the ability of a recipient cell to take a foreign DNA. It may be natural or it may be artificial. In a artificial transformation, there is a use of some treatment, use of low temperature, use of high temperature, use of solution like NaCl, calcium chloride, and this treatment can increase a permeability of a recipient cell, permeability of a cell wall of a recipient cell, so that the foreign DNA get entered in it. The transformation process occurs in a naturally Streptococcus pneumoniae and in a Haemophilus influenzae. In a Streptococcus pneumoniae, there is a involvement of a competence factor in a at time of uptake of a DNA, whereas in a Haemophilus influenzae such competence factor is not involved. The Streptococcus pneumoniae takes a DNA from any source, whereas Haemophilus influenzae takes a DNA of a same species. In Streptococcus pneumoniae, single-stranded DNA enters in a recipient cell. In a Haemophilus influenzae, double-stranded DNA enters in a recipient cell. In a cell of Streptococcus pneumoniae, the DNA binds to a cell surface and enters in a recipient cell, whereas in a Haemophilus influenzae, there is a formation of transformosome buds, which facilitates a DNA uptake and a penetration. So thank you very much friends. Please subscribe my channel and share my video to your friends.